Hello, thank you for your interest in my poster presentation. I am Eunmi Kim from MCPHS University in Worcester. I have been investigating the network modules identifying unique pathways for breast invasive lobular carcinoma. Breast cancer is one of the most deadly causes of death in women worldwide. It is classified into non-invasive and invasive, but with more clinical specificity, lobular and ductal carcinomas represent two breast cancers in terms of locations at the point of diagnosis. Majority of breast cancers occur in women due to genetic mutations as a result of the aging process and life in general, rather than inherited mutations. This study seeks to characterize age impacts on differential expression patterns between invasive lobular carcinoma, ILC, and invasive ductal carcinoma, IDC, and how protein-protein interactions and biological pathways associated with cancer progression are affected by those. Breast tumor sample RNA sequence and clinical data from Cancer Genome Atlas, TCGA, which is open to public, were interrogated. I separated them into IDC and ILC patients according to age. Using R as a tool, I found genes differentially expressed between ILC and IDC for younger group and old age group. Using these genes, I observed protein-protein interactions using the string network on a software called Cytoscape, and J-active modules in Cytoscape were also examined. These are the overviews of visualization tool Heatmap. I found 1,721 differentially expressed genes between ILC and IDC in patients 50-year-old or younger on the left side, and 126,075 genes in all age group patients were depicted on the heat map on the right side. The red dots represent the upregulated genes in one phenotype to another, and blue represents downregulated genes in this phenotype compared to another phenotype. These are the protein-protein interactions found in both groups. Cytoscape allowed me to discover numerous protein-protein interactions depicted as nodes and webs. Above all, I found bromodomain containing protein 7, BRD7, to be most interesting in the middle of the network, interacting with other neighboring proteins. It is downregulated in the ILC as a tumor suppressor gene, indicating ILC to be more aggressive than IDC. Similarly, you can see breast cancer gene 1, BRCA1, downregulated in ILC compared to IDC. However, I found cyclin-dependent kinase 1, CDK1, and SRC family protein kinases, a proto-oncogene, in all age group to be downregulated in ILC relative to IDC. These two genes promote cell cycle regulation and cell apoptosis, respectively. This made me conclude that more protein-protein interactions need to be investigated in all age group for more definite conclusion that ILC is commonly more invasive than IDC in terms of cell proliferation. From TCGA data, I found thousands of differentially expressed genes among different age groups. I saw the trends that protein-protein interactions between BRD7 and BRCA1 were upregulated in IDC, which lead to tumor suppression or cell growth checkpoint in the younger group. Therefore, I confirm the tendency that ILC is more aggressive than IDC, but still with inconsistency across the age groups. But I hope that the further analysis of differentially expressed genes among various age or racial groups could be conducted in the future. identifying the most significant genes causing malignancy to be targeted in the treatment of breast cancer. This is my reference. In the end of my presentation, I was Eunmi Kim, and thank you very much for listening.